We're just having a few issues with the Zoom, so we're trying. They have visual but no audio, so we're trying to fix that, and then we'll start shortly.
They have auto now? Okay.
Okay. Perfect. Okay, as we're a few minutes behind already, I think we'll get started. I would like to thank you all today for attending this Rising Star seminar series. This is a IDI seminar and host events microbial biology thematic program event. This is a seminar series we've had for the last, this is the third one that we've had, I believe. And the idea of the Rising Star seminar series is, instead of inviting more established, older professors, here we'd like to highlight investigators that are on earlier in their career, mostly assistant professors that are on a strong trajectory and doing some exciting work. And we also want to pair this with career events. So today at 4 o'clock, there's a career seminar discussion with Dr. Dana, who's going to talk about his career path. We want to highlight the diversity of different kinds of faculty within our departments and around the country, and show that trainees in our departments that there are roles for them in academia and academic positions accessible to them. So with that, I'm going to introduce Dave, who's the host for today's visit, and he's going to introduce the speaker. Thanks.
Thank you. So hello, everybody. I'm David Mackey. Thanks for being here. It's really my pleasure to introduce Dr. Dana, Christian Dana, or Colo. I hadn't met Colo personally until yesterday, but we had a really lovely afternoon and dinner, and it's been a pleasure to get to know him. I knew of his science. So a bit about him. He got his bachelor and master's degrees at University of Misiones in Argentina. Did I say that right? And then went on to get his PhD at University of San Martin in Argentina also. Then he moved to Harvard, where he postdoced with Brett Ossibel. From there, he moved on to a position at University of Virginia, where he's currently an assistant professor. He, in 2020, got a career award from the National Science Foundation, and in the years from around then up to present has been publishing some really excellent papers focusing on the way that plants control the partitioning of metabolites between their intracellular space and the apoplastic space where pathogens are located, and how control of access to those metabolites, particularly amino acids, really influences both the nutritional status, but really, I think, primarily the virulence expression of the pathogen. And I think that's largely what he's going to talk to us about today. So join me in welcoming Dr. Dana.
Thank you. Should I use this microphone, perhaps? This? Yes. Can you hear me? Yes. Okay. First of all, thank you so much for inviting me. It's a great pleasure to be here in a large community of plant research people, which is not very abundant in UVA nearby my lab. So it's a great pleasure to be here. It's also a special treat for me because when I was in grad school finishing my thesis in Buenos Aires, I developed a very profound respect for Dave's work from the years when he was in Jeff Dangle's lab, and we never met until now, and I hope that it's going to be a fruitful interaction from now on. Okay, so I understand that the audience is broad, so I'm going to give a broad introduction because many of you may have an interest in human immunity or health in general, and some others are more plant specialized. So please bear with me, don't fall asleep if you're a plant specialist. I'm going to show a few introductory slides to frame the work that we do in my lab, and then we will go directly into the hard science and the major findings that we have done in the past few years. All right? So plants represent the vast majority, the large majority of the biomass in our planet. This is a plant's planet, I would say. And I would recommend all those who are good fans of nature to watch a BBC documentary that came up in 2012, which is called How to Grow a Planet. It's an amazing documentary about how plants change and made, changed the planet and made life actually in our planet. And I picked this picture because this is the Blue Ridge close to our home, town in Charlottesville. And you can see how abundant plants are in our environments. All right. So why worry about studying plants? As we all know, the growth population that we have seen in the past 150 years, especially the last 50 or 60 years, has no precedent in history, as far as we know. We are reaching a world population of 8 billion people, right? And this is bringing conditions that are extremely challenging to the planet and to ourselves. And there are very good examples from history that we can use to have a sense of how important it is to make sure that we are able to grow food even in conditions that are very disfavorable. Some of the examples come from extinct civilizations. So these are all civilizations that, well, except for the Irish potato famine and for the contemporary Somalian people who live under very rough circumstances, all these are postcards from the past, right? Here we have the Sumerians, the Egyptians, here, the Sumerians here, we have the Mexica people, and more recently the Angkor Wat civilization, all went extinct, probably due to a combination of climate change and political instability, war, and stuff like that. And all this happened in circumstances in which the planet, the human population had very, very low numbers, right? So it's hard to imagine what may happen now with this amount of people living in planet Earth if massive climate change starts to affect food security. So I think that we need to pay strong attention to this problem and make sure that the plant sciences are well funded. Okay, so in case you have not paid attention, when you go to the wild, most of the time you encounter plants that are perfectly healthy, right? These pictures depict plants, tomato. This is Arabidopsis, the model plant that we use in my lab. But here we have seedlings, barley seedlings, and other plants, and they all look happy and healthy. This is by far the default state in which plants live in their environment. And when you think of the environment itself, it becomes really challenging to understand how is that they maintain themselves in such a healthy state, being surrounded by microbes. They cannot walk, they cannot change places. They have to do everything through biochemistry, right? They are born in the exact same place where they were to spend their entire lives, in the place where they germinated, and they have to endure whatever situations happen in the environment. Some trees in the West Coast are by now, as far as we know, almost 4,000 years old, and they are still holding strong and adapting to whatever happens to them, right? So in particular, we worried about and we care about how plants prevent infections and stay healthy in environments that are normally surround or full packed with potential pathogens. One thing that we learned in the past 20 years is that plants have evolved very sophisticated system to perceive molecules that are unique to microbes. I'm sorry, I'm in the middle of your view here. See. So they have evolved very sophisticated mechanisms to perceive flagellin to perceive transcriptional components of the translational components of the ribosome of bacteria, cell wall components of bacteria, and the same is true for viruses and fungi. In Arabidopsis, in the model plant that we use, there are encoded about 600 receptor leucine rate repeat receptor kinases and 57 leucine rate repeat receptor-like kinases. These are receptors that are expressed by the plants, and they localize to the plasma membrane for the most part, where they patrol the environment, the extracellular environment, for potential incoming microbes. Just to give you an example, the equivalent to these innate immune receptors in humans is a very low number. It's a very low number. It's 11 members of this family in humans. In mice, there are 13, right? In Arabidopsis, there are 600, and in monocots like rice or wheat, they are about 1,000 or 2,000 in some cases. So it seems very tempting to speculate that by expanding these families that allow plants to perceive specific components from microbes, they may compensate for the lack of an adaptive immune system that we have in invertebrates, right? So I want to introduce to the idea that plants make organic molecules from inorganic elements that they find in the soil and carbon dioxide that they take from the airspace. With that and sunlight, they drive the synthesis of sugars, and also most of the amino acids are synthesized in the photosynthetically active cells of the plants. When we think of plants, we tend to believe that plants are self-sufficient organisms entirely. And that is not true. The truth is that the photosynthetic cells, the truly photosynthetic cells that plants use to generate most of the nutrients that they need, are located in the mesophyll of the leaves. The rest of the cells in the body of the plant are dependent on these photosynthetically active cells to obtain nutrients in the form of sugars and amino acids. And those sugars and amino acids that are synthesized in the photosynthetic cells of the leaves are typically uploaded to the phloem system depicted here in red, and they travel to distant parts of the plant to nurture cells that cannot make their own food. This opens a very interesting opportunity for pathogens to take advantage of this trafficking of nutrients. When we look at a cross-section of a leaf, what we see is an upper epidermis, a lower epidermis with openings that are the stomata, right? Then we have photosynthetic cells here, very compacted, which are called the palisade. And then we have this spongiform tissue where the infections normally happen. Most of the leaf colonizing bacteria, which is what concerns my research, are able to colonize the nutrient and water-rich fluids that wash the cells in the mesophyll, in the spongy mesophyll. Why is that? Well, why is that there are nutrients and so much water in these spaces around the cells? Well, the reason is that in Arabidopsis and many crops, dicot crops, all the sugars and amino acids that are synthesized in photosynthetic cells need to be exported to this intercellular space as a first step in transferring them to long-distance tissues. So once they reach the leaf mesophyll, then they can be uploaded into the vascular system and travel long distances to nurture other tissues and cells. Okay, that's why there are so many sugars and amino acids in this space, and that provides a very a unique opportunity for pathogens to take advantage of these resources. I want to highlight amino acids because the rest of my talk is going to center on that. The amino acids that we find in the mesophyll, in the apoplast space of the mesophyll cells, is very rich, as I said, in nitrogen-containing and carbon-containing amino acids and sugars. And many of the amino acids and sugars that we find in the mesophyll of the leaves, in the apoplastic space, are able to support the growth of bacterial pathogens. In this case, what we see here is the growth of Pseudomonas syringae, Patovar DC3000, tomato DC3000. This is a pathogen of Arabidopsis. So this bacterium is able to use many of these amino acids that are depicted here in different colors and the sugars, right, as the only carbon source to support growth in minimal medium. That obviously shows that they are able, metabolically able to use any one of these metabolites, plant-made metabolites, to support their own growth. So again, the availability of these metabolites in the leaf apoplasts provides a huge opportunity for the pathogens to establish an infection. The mechanisms by which the composition of the leaf apoplast is controlled, happens mostly, it's mostly dependent on the activity of transporters, right? None of the metabolites that are made intracellularly are going to go out of the cell into the leaf apoplast across the plasma membrane by osmosis or wherever. They will have to use transporters. So depicted here are some transporters that are able to move amino acids from the cytoplasm to the leaf apoplast of the leaf. And depicted in the middle here with an inward arrow, we have an influx transporter that is able to bring back some of these amino acids that are secreted into the apoplast, bring them back to the cytoplasm, right? Those inward transporters are going to need protons to co-transport amino acids. As you may know, the pH of the leaf apoplast is typically 5.7, and the cytoplasm is close to 7. And that is because of the activity of this proton pump ATPase complex here, depicted here. This complex hydrolyzes ATP and pumps protons out into the apoplast. And those protons are used by these influx transporters to co-transport amino acids back into the cells, right? So at any given time, the concentration of amino acids that we find in the leaf apoplast is going to depend on the export through the efflux, which is mostly passive, and the import through the influx transporters that is mostly active, right? So again, this provides a huge opportunity for bacteria. The only problem for bacteria to gain access to these resources is that these cells are carpeted with receptors. Many of the receptors that I mentioned in one of the first slides, which are the pattern recognition receptors, are expressed and localized at the plasma membrane, and they are like booby traps. Whenever a new microbe enter the leaf internal space, the leaf apoplast, they will, in a way or another, trip into one of these pattern recognition receptors, and they will trigger immune responses that will eventually make their lives more complicated. So again, this is a very nutrient-rich space. It's wonderful for the bacteria, but they have to learn how not to trigger these immune responses, or eventually how to suppress them if they happen to trigger them in the first place. All right. For some time, we have known that composition of the leaf apoplast has an impact in the way the bacteria infect the apoplast. We know that when immunity is elicited with one of those very conserved microbial proteins, for example, that we call MAMPs, they trigger an immune state that we call pattern trigger immunity, right? Some pathogens have learned to suppress this pattern trigger immunity very, very early in the interaction, right? And that is exactly what makes them pathogens. They are microbial pathogens because they can do this. This is a very, very, there are very unique cases in which microbes evolve into pathogens and are able to do that. For the most part, plants are resistant to microbial infections, right? And this is because they elicit immune responses that cannot be suppressed by the pathogen, and then the plant suppresses the infection very early in the interaction. Okay, so this is a model to introduce the concept of how this pattern trigger immunity works. So if we imagine an infection here in the mesophyll of the leaves, this is a magnification of one of these photosynthetic cells. What we would see is that they express different flavors of these pattern recognition receptors that are going to be able to perceive different patterns that are expressed by microbes. When the microbes come in this case, a gram-negative bacterium, they will trip into one of these receptors, and they will elicit responses that are going to be mostly mediated by salicylic acid, which is a very important mediator of immune responses in plants. That is going to trigger extensive changes in the physiology of the plant and the transcription programs that ultimately is going to, excuse me, this is not the slide that I was supposed to show. Okay, I will use it anyways. Just give me a sec. This is the slide that I was supposed to use. So they come in, they trigger the immune responses. The immune responses are going to suppress the growth of the bacteria very early in the interaction, right? And as a consequence of that, we're going to see that the infections don't progress and the plants look healthy, right? The question is, how is this happening? And this is what we focus on in my research program. Until recently, we have very few ideas of how this happened. We knew that changes in the composition of the leaf apoplast may contribute to this phenomenon of suppressing the growth of the bacteria, but we didn't know by which mechanisms that happened. So let me go back one slide. So, okay. So to study this phenomenon, what we do is we use a purified peptides that we call flagellin 22. It's a 22 amino acid synthetic peptide that contains the minimal epitope of the bacterial flagellin. We use this as a mean to induce immunity in the plants. So by all means, this process, this phenomenon works as vaccination. So when we expose the plants to this peptide, they perceived the peptide via one of these specific pattern recognition receptors, right? And they mount an immune response. 24 hours later, so there is no pathogen there, right? So these plants are free to go on establishing a very robust immunity without the interference from the pathogen. 24 hours later, when we ran an infection on the same leaves, what we see is that the plants that were mock treated, they develop a very robust infection. But the plants that were pre-immunized with flagellin 22, they develop no symptoms. And when we take samples from those leaves, and count the colony-forming units to establish infection titers, what we see is that the plants that have been immunized with flagellin are significantly, these are orders of magnitude here in colony-forming units, significantly more resistant to the infection than the MOC-treated plants. All right, so we will use this type of assays quite along the road in this presentation. These differences in CFU counts resemble, in a way, what happens when plants are inoculated with non-pathogenic bacteria versus bacteria that can produce a very robust infection. So it looks like the pre-immunization turns or is able to turn pathogenic bacteria into a non-pathogenic like bacterium. Okay? Okay, let's go through this. This slide is just to remind you that there are pathogens that can suppress this type of immunity. And they do that. Sorry, this was the previous slide. So they do that by expressing a protein complex that is called the type 3 secretion system that is able to inject effector molecules into the cytoplasm of the host cells. And many of those effectors are going to have an impact in the signaling of this PTI immunity, right? And are going to suppress immunity. And with that, the pathogen will proliferate and establish a very robust infection. I want you to hold in your minds for a second these molecules, these type 3 secretion effectors, and perhaps coronatine, because we're going to go back to that at the end of the talk. Okay? We're going to figure what is the impact of immunity on the expression of these systems, of these pathogenesis systems. Okay, so again, the question is, how is this immunization helping the plants to resist infections? Okay. Again, this is a slide that is just to remind you that when we immunize plants and then infect them, we don't have symptoms. And again, the CFU counts are very low compared to the MOC controls. All right. And again, we know that the apoplastic space of the leaves is very rich in nitrogen, in amino acids, many of which are able to sustain very robust growth, bacterial growth in vitro, right, as a sole source of carbon and nitrogen, right? So we started to, by investigating how is that the concentrations of amino acids in the leaf apoplast change in response to the immunization of the plants with the flagellin 22 peptide. And what we found is that a very large number of efflux and influx transporters, again, the ones that control the concentrations of amino acids in the leaf apoplast, are up or down regulated by flagellin. Remember, this is not a pathogen, it's just a molecule that elicits an immune response. And just by doing that, that molecule is setting off a program that changes the expression of many of the transporters that are in charge or potentially in charge of defining the composition and the concentrations of amino acids that are available in the leaf apoplast. So we started to ask ourselves, what is the impact? Is there any impact in the amount or composition of amino acids in the leaf apoplast when the plants are immunized with flagellin 22? And the answer is yes. So what we did for these experiments is to grow our Arabidopsis seedlings in liquid culture. And we did that because as they grow, they exude metabolites into the medium. And though the composition of the medium after 10 days is quite similar to the composition of the leaf apoplast that we find in a normal leaf, right? And the reason why we do this instead of looking directly into the composition of the amino acids into the leaf apoplast is because extracting apoplasmic contents from a leaf is extremely challenging, as opposed to this method that allow us to obtain very large amounts of exuded metabolites into the liquid very easily. So by using by assessing the composition of the exuded metabolites, we have an estimation of what is or what should be happening in the leaf apoplast of an adult plant. So what we do is we collect the sample, the liquid samples, right? Originally, the medium doesn't have any amino acids or any of the sugars that we find 10 days later. So all the amino acids and all the sugars that we find in the liquid here are coming from the plant. Those are plant-made metabolites, all right? What we see is that the plants that were mock treated have, an X amount of amino acids. This is micromolar. But the plants that were immunized with flagellin peptide, they have almost double the amount of total amino acids in the liquid, right? And at first we thought, well, this doesn't make any sense. Because I've been telling you, well, this bacteria love to use these amino acids as a source of carbon and nitrogen. So the more they have, right, probably the more they will grow. So how is this going to explain how is that flagellin immunization actually suppresses the infection with the bacteria? All right, so we started to scratch our heads, and we said, well, is this really, due to transport? Is something related to transport in the seedlings that is contributing to the increase of amino acids in the medium? And the answer is yes. When we do uptake of radio-labeled amino acids in the seedlings, what we found is that the uptake in the seedlings that were immunized with flagellin has dropped below 50% of the uptake level in control seedlings, right? So yes, the excess of amino acids that we find in the liquid, let's say the extracellular amino acids, right, is probably a consequence of the shutdown of uptake, which is happening all the time in the plants. Okay, remember the composition of the leaf apoplast, and in consequence that of the medium of the seedlings is again controlled by the secretion rate and the uptake rate. Those two processes are going to define at any given time how many amino acids we find outside the cells or outside the seedlings. So what we found again is that we use 3 different label amino acids. This doesn't seem to be a specific inhibition for the uptake of any one amino acid in particular, but it seems to be a broad response. It looks like the uptake of amino acids is being shut down by the immunization, right? What you have here is the amount of labeled proline that remained inside the seedlings, okay? So in normal conditions, in mock, you have this much labeled amino acids inside the seedlings, and for the elicited seedlings, you have significantly less. That is a good indication that the uptake is being compromised. Consistently with this inhibition of uptake, what we find is that the exudates are very rich in these 3 amino acids, among many others. So there is perfect correlation between suppression of uptake and the increased concentrations of amino acids in the exudates of the seedlings. And to figure if this is a consequence of breaking down the concentration gradient of protons across the plasma membrane, remember that the influx transporters typically work by co-transporting protons and amino acids into the cell. So if you break that differential potential across the plasma membrane, you can break the influx of amino acids. And you can have a phenomenon that looks like this. And this is exactly what we found when we treat our seedlings with a chemical that breaks the differential potential across the plasma membrane. What we have is the accumulation of extracellular amino acids in the medium. Okay. So far, this seems to indicate that immunization with flagellin, and not only with flagellin, this also happens with at least two other MAMPs that are specific microbial specific molecules. It seems to be a generic way of responding to the elicitation of immunity. One of the first things that they will do is to shut down uptake. We do not know if this also applies to sugar, but we suspect that that is the case. It definitely applies to amino acids. Okay, but again, this doesn't make sense because we would have expected exactly the opposite. We have, since bacteria are so happy growing in these amino acids, how is that, offering them more amino acids is going to make any good, right, for the plant? So we kept on scratching our heads just to demonstrate that this phenomenon is receptor dependent. I want to show you these slides. So the data here comes from receptor mutants that are unable to perceive either flagellin or a second MAMP called EFTU, right? And what we see is that the drop in uptake elicited by the immunization is specific is receptor specific, right? This is not an artifact of any kind produced by the way in which we grow the plants. We know that if the receptor is not there and flagellin is not perceived, the seedlings are blind, they don't see and they don't express any changes. And the same is true for this second pattern recognition receptor. So this seems to be a general theme across immunized plants. So how does the inhibition of uptake in the apoplasm metabolized bacteria affect the infections in the leaves? And so to address this question, what we did, we cannot assess directly uptake in intact leaves. So what we have to do to assess uptake in leaves is to obtain cells, right? So we obtain protoplastic cells from the mesophyll cells that are photosynthetically active. And we did the similar uptake experiments that we have done earlier with the seedlings. And so we isolate these cells, right? We remove the plasma membrane and we offer label amino acids. And right, we offer label amino acids. The cells are going to uptake some of those amino acids as they normally do, right? And that's how we assess uptake activity. Then we count radioactivity in a liquid scintillation counter. And what we find is that the protoplast cells that have been mock treated uptake this much amino acids, while the protoplasts that have been immunized with flagellin do not uptake that much. So that indicates that what happens in the seedlings is also probably happening in the protoplast, in the mesophyll cells of the leaves. So we run a second experiment to further understand how is this happening in the leaves, in the intact leaves. We immunize our plants as we normally do, right? And we obtain apoplastic washing fluids from the leaf apoplast, and we quantified amino acids in the apoplast of intact leaves. And what we found is that the amino acids accumulate over a period of 24 hours. So it looks like the same thing that we see in seedlings is happening at a slower pace, probably, in the adult plants. Remember, the increased concentrations of amino acids in the seedlings can be seen only 4 hours after the treatment. In this case, at 4 hours we don't see a major effect, but we do see a very clear effect at later time points. Now you have to remember that in order for immunized plants to become resistant to infections, we have to let them undergo the normal immunity program for 24 hours before we infect them with a pathogen, right? So by the time that we infect these plants with pathogens, the plants already have double probably the amount of free amino acids that are available in the leaf apoplast. And again, this doesn't explain anything. It's exactly the opposite to what we would have expected, right? Because that seems to indicate that these bacteria that are incoming here are going to be happy with more amino acids and potentially going to be able to produce a more robust infection, okay? When we profile via LCMS the amino acids that we recover from the leaf apoplast, many amino acids were enriched in the flagellin-treated samples. So we decided to co-infiltrate bacteria with these amino acids in order to mimic the situation that they would encounter when they are trying to produce an infection in a pre-immunized plant. Okay, so what we did is we co-infiltrated bacteria with any single one of these amino acids, right, in naive plants that have never been treated with any challenges, right? And what we found is that glutamine, serine, and valine are able to strongly suppress the growth 24 hours later, right, when co-infiltrated with the bacterium. So we said, well, this makes sense, right? So perhaps these immunized plants are flooding the apoplast of the leaves with amino acids because that is a good thing. How? We don't know, right? But if we do mimic this system by just co-infiltrating the bacteria with purified amino acids, we see the same. So this seems to fit perfectly, but still we do not know how. Okay, so we went back to the literature, and there are beautiful papers that came up in 2018 and 2019, some of which indicated that immunized plants are able to suppress virulence in the pathogen. So what you see here is a heat map of gene expression of this Pseudomonas syringae bacterium that were recovered from plants previously treated with water, the mock samples, or treated with flagellin, the immunized plants. And what we see here is the ratio, right? So in red, you have overexpression. These are geo terms, categories, right? And what these people found is that in the flagellin-treated plants, the bacteria are expressing very, very low levels of genes encoding type 3 secretion system and chromatin biosynthesis genes. Remember for a second that type 3 secretion systems and type 3 secretion system effectors and chromatin, which is a small molecule that this bacteria produce, are the virulence factors that these pathogenic bacteria normally use to suppress immunity very early in the interaction, right? So the fact that these bacteria are having a hard time expressing virulence may per se explain how is that they are growing less in the immunized plants. So we thought, well, maybe our amino acids are having an impact in virulence of the plant, right? And perhaps what our assumption that offering them more amino acids would make their life easier because they can use them and grow to higher titers is completely wrong because perhaps, right, those amino acids, what they are doing is actually suppressing the expression of virulence and compromising the infection, right? And this is exactly what we found. So remember that when we co-infiltrate plants with glutamine, serine, or valine, the counts, the bacterial titers are significantly lower than the controls. And what we found is that when we use these three amino acids, glutamine, serine, or valine, individually, right? to co-infiltrate them with bacterium and then recover the bacteria 3 hours after the inoculation, we assessed gene expression in the bacterium, and we found that indeed these amino acids were suppressing the expression of very key components, very key genes in the bacterium that are essential for the expression of the type 3 secretion system and the synthesis of this small molecule called coronatine. So we think that now things make sense. We think that what the plants are doing is to flood very quickly, shut down the uptake of amino acids to allow amino acids to accumulate in the leaf apoplast in a way that is going to compromise the establishment of an infection via suppressing the virulence factors that the bacteria need to express in order to establish a very robust colonization of the leaf apoplast. All right. I'm going to skip this because I think this is, well, let's go to this final part. I don't know what time is it. How much time I have left? Nothing.
11:50.
Nothing. Okay. So, so what we found is that relay is an enzyme that synthesizes PPGPP, the alarmon that is very common throughout the microbial kingdom, bacteria in particular. So what we found is that in the pre-immunized plants, the expression of relay is also suppressed, and there are published studies in the mid 2014 and 2015 that indicate that the levels of PPGPP are in part controlling the expression of virulence, the type 3 secretion system in particular. So we think that by flooding the apoplast with amino acids, the plants are actually suppressing the expression of virulence via suppressing the synthesis of PPGPP by downregulating REL-A. So why REL-A is important? Well, this is from E. coli studies. In permissive environments, the levels of PPGPP are very low, and then sigma 70 is going to allow the transcription of genes that are pro-growth, right? In non-conducive environment or restrictive environments, PPGPP is going to change this balance and is going to allow other sigma factors to drive the RNA pol 2 to transcribe genes that are essential for virulence and survival, right? So if you have low levels of PPGPP, like here, in a situation where you are trying to establish an infection, that is going to be very bad for the bacteria. All right. So here's a model to understand how the whole thing works, as we understand it so far. So when a pathogen comes in, it may trigger one of these booby traps, one of these receptors. That's going to elicit a very fast change in the concentration of protons in the apoplast, and that is going to inhibit the uptake of the amino acids. Remember that in order for the uptake to happen, protons are necessary because the uptake is done via cotransporters. All right? So after that, everything goes down for the bacterium because amino acids are going to accumulate outside of the cells, right? They're going to reach the bacterium, are going to suppress relay, which in turn is going to suppress or prevent the onset, I could say, of the expression of virulence in this bacterium. And in the meantime, the plant is going to continue with its immunity program. It's going to start expressing antimicrobial molecules like lignin, for example. And by the time that the bacterium reacts to this trickery, I could say, it's probably too late, and they can no longer suppress the responses, and it's too late to establish an infection. All right? So I'm going to go straight to, I'm sorry, I don't know how to pass this. I'll go straight to the, some of these are slides that I would like to show Dave at some point later. So let's go to the acknowledgements. These are Shamu Sang and Pramod, together with Patrick, contributed mostly to the uptake experiment and the one of the studies. Renyo Lee and Phil contributed to many other aspects of the data that I showed today. Many of them are graduated by now and looking for jobs. So if you if you need postdocs that are trained in these type of things, drop me an email. All right. Thank you so much.
Are we going to open for Zoom, or will you? There are any questions here. Go ahead here. Maybe send a chat that says.
Yeah, go ahead.
Do the bacteria metabolize remain serine less effectively than other classes?
No, they love them. Those are the amino acids that they prefer. So these pathogenic bacteria in particular have evolved to be very effective at using the amino acids that are normally available in the apoplast of the leaves, among them glutamine and serine.
So it seems that a bacteria that can evolve resistance to sensing
Yeah, you could think so. So we have some evidence for valine in that sense. We have a study that I didn't have time here to show. It's a metabolomic. It's a metabolic modeling study that we have done, indicating that yes, they have mechanisms to hydrolyze valine in particular as a way to relieve the inhibition of the suppression. Does it make sense?
Right. So those
We don't have any overexpressors for those mechanisms, but we presume that that would be the case. Yeah. Yes.
In a larger plant like a tree, say
Yeah.
a sugar maple tree, can a response in one part of the tree be conveyed to another part of the tree?
Yeah.
Or is it very localized?
Yeah, that's a very good question. So there are systemic, responses that have been described in the past 20 years. I think that they also apply to trees. They do apply in Arabidopsis. You can elicit different responses in one leaf, and then you find out that distant leaves have also acquired, have become more resistant to infections.
Question was, how is that transmitted?
Yes. So typicolic acid is one of the acids that travels through the phloem and seems to play a major role, going from challenged leaves towards non-challenged leaves and elicit this type of responses. But there are also volatiles like methyl jasmonate and methyl salicylate that are able to communicate stress even across plants, not only within plants, but also across plants. Does it answer your question?
Yeah, well, so is it then maple syrup? That's how I got to this.
Yeah, I don't know. That's a good question. Yes.
I have a question. Maybe I missed this, but do you know anything about the mechanism of suppression and why these particular amino acids? Are there others that can also do this?
No. Yes. We do not know. This is something that is in the makings. We are trying to understand. It seems that different amino acids have different impacts. Some amino acids don't have any impact. But for example, in some of the slides that I couldn't show, we have evidence showing that other amino acids are actually extracted at later time points in the infection by the bacterium, and those happen to have a virulence-inducing effect. Glutamic acid, for example,
So it's interesting this PAMPs can reroute the transport activity on the plasma membrane. I'm just curious whether similar events also apply to intracellular pathogens because they also produce MAMPs.
So I don't know, but I can imagine that by dissipating the proton gradient across the plasma membrane, vesicular trafficking is also going to be impacted. So maybe that's a way to prevent intracellular pathogens from coming in to the cells. Does it make sense? Yes.
Is there any effect of like the pH of the apoplast? You said it's
Right.
first thing is shutting down,
Right.
obviously proton pumping, so the pH is going to go
Yeah.
up. Does that influence virulence?
Right. So flagellin has been described more than 20 years ago to elicit the transient alkalinization of the medium where you grow the seedlings, for example, as I show in some of my slides. There is a very transient increase in the pH after you add flagellin into the medium. So yes, the two things probably go together. It could be that the alkalinization of the medium of the apoplast is probably the consequence of breaking the proton, shutting down the proton gradient across the plasma membrane. But we do not know if that happens in the apoplast of a leaf, right? We try hard to measure very subtle and quick changes, and we didn't succeed. Yes.
So you're talking about vaccination.
Yes.
I like that. You're talking about vaccination.
Right.
So that brings in mind two questions. One is how long does the response of flagellin last? And the second question is, now there's at least in a million immunology, there's a concept of trained immunity, meaning that you can have by exposing, repeated exposure to pump, etc. You can change the innate response. So the question now is, when in plants, if you continuously expose them to flagellin, can you have trained immunity?
So I think there are papers dealing with this problem, and they found out that the immunization lasts for a few days, like 4 or 5 days. So it's not a permanent situation, right? If you do one immunization after the other, after the other, I suppose that it's going to be hard because the cells may end up just dying. Because there are many things that are happening, not only this within the cells. Yes, Dave.
I have one. It's, I guess, less of a question and more of a comment, but I was wondering if you could just speculate about how these mechanisms relate to endophytic microbes that are not pathogenic. I mean, mostly the bacterial population they're presenting them there all the time.
Right.
How does this distinguish, say, of a pathogen as opposed to all the other microbes that are there, and how do these mechanisms affect the abundance of the endophytes?
Right. That's $1 million question. Yeah. So I don't know. I mean, no, I think that we nobody has a definite answer. We are just starting to understand how that works. One thing that we found and we published recently is that the levels of amino acids in the root exudates, for example, they have a major impact on making associations with beneficial bacteria, for example. But the cases that we found is that an excess of amino acids in the rhizosphere, for example, is detrimental to the metabolism of the bacterium, and that kicks back by not improving the growth of the plant, which otherwise could happen. Intracellular symbionts or I don't know. These bacteria that we use, they live in the rhizosphere. They don't go in.
Yeah, so in mammals, of course, we see a lot of specialization of immune cells. So I'm curious, in plants, do you see these specialized cells or a conversion of cells to some sort of specialized function? In particular, I was interested in phagocytes.
Right. Well, I mean, there are no circulatory cells that we know of in plants. So there is nothing like a T cell or a B cell. There are no antibodies. These type of responses are cell autonomous responses for the most part. Every cell in the plant body is supposed to express, a suite of different pattern recognition receptors, and are supposed to be well armed to respond and defend themselves individually against these challenges. So I don't know. There could be some type of specialization that we haven't discovered yet. All right.
There's no other questions. Let's thank Dr. Dano one more time.
And thank you again. Thank you so much for making the time. I know this is summertime, so what are you doing here?